Hello everyone. Let's get started. Today we're going to be talking about the regulation of gene expression. I've made a list of the goals of this lecture for us so that as you're studying this material, you'll know which points of the lecture to focus on. The first thing we're going to talk about is some of the vocabulary around gene expression. We'll talk about what are referred to as global mechanisms of regulation, and we'll focus specifically on the global regulation of transcription as a portion of the gene expression process. We're going to talk about global regulation of transcription, both in terms of access to the gene itself. In other words, whether or not the gene gets used, expressed, in other words, and access to the regulatory regions of the gene, or what we sometimes refer to as fine control mechanisms. Towards the end of the lecture, we'll touch on a couple of the other ways outside of transcription that genes are regulated by, and some of the few other cellular control mechanisms. But we're really going to focus today on these global regulatory mechanisms of the portion of gene expression that is transcription. Now remember, when we talk about gene expression, we're talking about two of the three statements of the central dogma. That DNA is transcribed to produce this molecule called RNA, and that RNA is then translated to produce proteins. That's what we refer to as gene expression. Remember that some genes are expressed constitutively, and what that means is they're expressed constantly or around the clock. And that's because the cell has a constant ongoing need for the protein that is being produced from that gene. We often refer to constitutive expression as these genes just being on all the time. But most genes are not constitutively expressed. Most genes are tightly regulated. Another way to think of that is that most genes have to be turned on when they're needed, but otherwise they are off. The cell will transcribe and translate those regulated genes only when it needs that particular protein at that moment. Now, it's important to remember that when you hear people use terms like on and off in relation to genes, it's not really accurate. In reality, when we say that a gene is turned on, we're saying that the gene is being upregulated or up-expressed. And when we say that a gene is turned off, we're saying that it's being down-regulated. The DNA molecule that sits in the nucleus of all the cells in your body is a remarkable molecule because it contains all of the information that is needed to make all of the RNA and thus all of the proteins that would be needed to construct a new you. Sometimes people have the idea that there's a different DNA molecule in different types of cells. In other words, perhaps there's a particular DNA molecule that you would find in liver cells and a different DNA molecule that you would find in the cells of skeletal muscle, and so on and so on. In reality, the DNA molecule is the same in each one of your cells. And that means it contains all of the information needed to build another one of you. The thing is, the information to make a protein is only the beginning In other words, the presence of a gene doesn't mean that a protein is going to be produced from that gene. Some genes, for example, are only expressed during particular times in an organism's life, and then never again. For example, there are lots of genes in your cells that are expressed only while you are a fetus, only while you are developing. Those genes are then turned off, again, to use that very general language. They're turned off once you were born, and they're never to be turned on again. It's not that the genes go away, they're still present in your DNA. It's just that they are never expressed again. And that requires regulation. That requires the cell to take steps to prevent those genes from being turned on again. So, some genes are turned on at particular times in a lifetime. Some genes are used throughout the lifetime of the organism, but only periodically. Some genes in our DNA molecule are never expressed. Remember, you contain a lot of DNA that you inherited from your ancient ancestors, from organisms that lived on this planet hundreds of thousands and millions and even billions of years ago. That DNA didn't go away. It's still inside of you. You just don't use it anymore. You don't express it anymore. A great example of that is the gene or genes that code for the production of a tail. You have those genes. You just don't express them anymore. Your ancestors had a tail. Ancestral primate type organisms had a tail. And through evolution, as we lost our tail, those genes weren't lost. They were simply turned off. They were silenced. They were no longer expressed. So, when we talk about the regulation of gene expression, what we're really talking about is the way that cells can control if a protein is going to get made, and if it's made, when it's going to be made, and when it's made, how much of it is going to be made for each and every gene that is regulated. Let's just take a minute now and review the definition of a gene. Remember, there's two types of code in a gene. There's going to be a sequence of base pairs that codes for a protein. In eukaryotic cells, it's one gene, one protein. That's not how it is in some prokaryotic cells. Remember, some prokaryotic cells have what are called operons, and those are combinations of protein coding regions to make multiple proteins from one gene. But eukaryotes have one protein made from each gene. Genes also contain a sequence of base pairs that regulate the expression of that gene, that determine, again, if, when, and how much of that protein is going to get made. Remember that in the regulatory region of genes, there's going to be a promoter. That's the region that the RNA polymerase enzyme is going to look for and bind to. But there are also going to be other types of regulatory code, including regions that are referred to as operators and activators and so on. So, gene expression and the control or the regulation of gene expression begins with simply accessing the gene. And frankly, accessing the DNA. One of the what we refer to as global control mechanisms of gene expression is simply controlling whether or not a gene is accessible, because the DNA may or may not be accessible. Cells can control whether or not the DNA is even reachable in the cell. Remember, DNA exists as a double-stranded molecule inside a living cell, and it doesn't simply float free. The DNA double helix is wrapped around histone proteins, and each time it wraps around those proteins, as you can see in the image at the bottom of the slide, it creates this little subunit of chromatin that we call a nucleosome. Lots and lots of nucleosomes are created and then stacked up on top of each other in a very regular pattern, creating that molecule that we refer to as chromatin. It turns out that there are different structures to chromatin. Sometimes there are regions of the chromatin that are more tightly packed together, and other times the chromatin is less tightly packed. How tightly packed the chromatin is going to determine whether or not that DNA is even available for transcription. And this is the fundamental way that a cell regulates gene expression, by controlling access to the DNA. On this slide, you can see a drawing of how the double helix of DNA is carefully wrapped up into nucleosomes and then stacked to produce chromatin. You can see a depiction of the double helix on the right here. And then you can see that that double helix is wrapped twice around a ball of protein that's referred to as histone protein. Notice that every time it wraps twice, then there's a little strip of DNA, and then it wraps around the next histone core, and then there's a little linker, and then it wraps more, and on and on and on, producing all of these individual nucleosomes. The DNA that stretches between two nucleosomes is referred to as linker DNA. The histones are actually not one, but multiple types, creating this sort of ball of protein that the DNA wraps around. Don't worry about the individual names of histone proteins. That's not important. Just understand that this ball of histone protein is going to be wrapped twice with the DNA double helix, creating this subunit of chromatin that we call a nucleosome. So as you look through a type of cell in an organism, particularly a multicellular organism like a human being, as you look from cell to cell to cell, the genes that are being expressed are going to vary from cell to cell to cell. And that's because different cells have different functions. The type of cell that lives inside your skin, for example, is going to have a different requirement for protein than a neuron in your brain. So that means that the skin cell is expressing different genes than the neuron is. That means that transcription looks different in that skin cell. Translation looks different in that skin cell compared to the transcription and translation that are happening in the neuron. Each cell is only going to express the genes that code for proteins that that cell needs in that moment, only when the protein is needed. A good example of that is the gene that codes for insulin. Insulin, of course, is the hormone that helps regulate your blood sugar, and it's produced and secreted by cells in the pancreas. If you look at heart cells, they're not going to transcribe and translate the gene for insulin. They don't secrete insulin. They don't use insulin. The gene that codes for insulin is present in the cells of the heart. It's just not expressed. So when we think about the genes that are constantly expressed or constitutively expressed, we often refer to those as housekeeping genes. These are the genes that code for proteins that are needed around the clock. So these are the kinds of proteins that all cells use. For example, the enzymes DNA polymerase and RNA polymerase are needed by cells, all cells. The RNA polymerase enzyme is needed all the time because transcription is going to go on all the time. Now, different genes will be transcribed, but the enzyme that performs the process of transcription is going to be needed for all those different genes. So the RNA polymerase gene has to be constitutively expressed. It has to be expressed around the clock. The histone proteins I mentioned a moment ago, they're going to be needed all the time because DNA has to be constantly wound around histones and then loosened up and then wound and then loosened up. And those histone proteins have to be repaired and replaced around the clock to maintain that chromatin. But remember, most genes are not constitutively expressed. Most genes are under regulated expression. They are expressed by cell type and according to need. So it's these global regulatory mechanisms that we'll really focus on today. These are the mechanisms that control when these regulated genes get expressed. Now, when I use this term global just means that it's a mechanism that is performed the same way across all different types of cells. In fact, across all three domains, bacteria, archaea, and eukarya. Everybody does this the same way on our planet if it's a global mechanism. And I said a moment ago that these global regulatory mechanisms all begin with just plain old access to the DNA. A gene can be regulated by controlling whether or not the DNA is even accessible in the cell. And that refers to whether or not the chromatin is tightly packed or loosely packed. If it's tightly packed, we call it heterochromatin. If it's loosely packed, we call it euchromatin. Heterochromatin is defined as tightly packed chromatin and chromatin that is transcriptionally inactive. In other words, the chromatin is so tightly packed that the genes that are in the DNA in that region of the chromatin are simply not accessible. The enzymes that would transcribe those genes are simply unable to get to them because of that tight packing. Euchromatin is different. Euchromatin is defined as loosely packed, exposed nucleosomes, and therefore transcribable DNA. Cells have the ability to control which regions of the chromatin are in the heterochromatin state and which regions are in the euchromatin state. There are mechanisms in place to move chromatin between those two states. If you look at the image on the right-hand side of the slide, Up at the top, you can see a representation of a nucleus, and it's been cut in half so that we can see the chromatin inside. Remember, through most of the life of a cell, the DNA exists in this chromatin state. It's only when a cell is preparing to divide that the chromatin condenses and becomes chromosomes. But it turns out that if you look at the DNA when it's in the chromosome form, you can actually visualize where the heterochromatin is and where the euchromatin is. It's fascinating. Notice how this chromosome, which has been replicated, so this is a pair of chromosomes, notice that it looks like it's striped. Everywhere that you see a dark banding on the chromosome, that is DNA that is in the heterochromatin form. Everywhere that you see a light colored band, that represents euchromatin. Now, on the bottom portion of the drawing, you can see an artist's representation of heterochromatin and euchromatin at a much higher magnification. You can see these blue balls that represent balls of histone protein, and you can see a black strand that represents the double helix of DNA. Notice that it's wrapped twice around each ball of histone protein, with linker DNA in between. On the right side, this represents heterochromatin, or the type of chromatin that is so tightly packed that it is transcriptionally inactive. The cell cannot transcribe any of the genes in this region because it's so tightly packed. Over here, the nucleosomes are much farther apart from each other. This is what we refer to as euchromatin, and this type of chromatin can be transcribed by the cell. So cells have the ability to go back and forth between heterochromatin and euchromatin in their DNA molecule in order to regulate which DNA is even available for transcription. I mentioned the insulin gene a few moments ago. The insulin gene would have to be in a region of the chromatin that is in the euchromatin state in the pancreas, so that the insulin gene would be available for transcription. In that heart cell, though, the DNA in the area of the insulin gene would be in the heterochromatin state, so that it could not be accessed for transcription and translation.